You ready?
Yes.
Okay, so we left off at metaphase yesterday, so let's do this. We're still in stages of mitosis. You got it? Yeah. Okay, we got metaphase. Now prophase had the most things happening with it, so there won't be, it'll speed up a bit right now anyway. So draw your cell. Now your chromosomes are going to be in, they're going to be in that double chromosome thing. So it's easiest to kind of make like a little X, if you will. Okay, I'm going to do, we only were doing 2 pair yesterday. And in the middle of each one, draw, look up here, draw like a little dot. It's kind of hard to see up here. That's the centromere that's holding them together. So we got 2 number 1s we'll do, and then we got 2 number 2 chromosomes. Put my little dot in the middle of those. Where are centrioles? The poles, the ends. Okay, so here's what's going on with this one. Double chromosomes. Line up in the middle. So metaphase, if you want to do this, I'm going to draw a little arrow to here. Meta means middle. Kind of easy to remember too. Meta, metal, lines up in the middle. So they line up in the middle. The second thing that happens is the spindle fibers attach. So go ahead and draw 1 spindle fiber. It's more than this, but we're just going to do it for our own purposes. 1 to each of these double chromosomes on each side. Now here's the one thing I put on here, as far as mutations and examples of things that can happen. Right now, UV can break, UV or any other environmental factors, UV can break spindle fibers.
So how often do cells have problems like this?
I don't know the number. Here's what I do know. That the mutation rate, and it does happen, it just does happen, because you got 50 to 100 trillion cells in your body. At any given time, this is a Nate math problem. At any given time, 2%, I can already do it. At any given time, 2% of your cells are going through mitosis. So that's 1 to 2 trillion of your cells, okay, are going through mitosis at any given time. That's times the 3 billion base pairs of DNA in each one of those cells. So right now, 1 to 2 trillion of your cells are going through mitosis, and right now, on average, any number of those is trying to replicate all 3 billion base pairs in each one of those 1 to 2 trillion.
So does it happen?
Huge numbers, huge numbers. They know this, that the error rate that happens, because DNA has so many little fail-safes in it that can fix each other, the error rate is less than what happens with computers. And I forget the numbers. It was like in 1 in every trillion calculations of the computer, there's an error. So something like in 1 in every 100 trillion in DNA, there's an error, or something like that. But the error may be in the junk, or maybe in a gene that's not used because you have it happening in a skin cell and not in a stomach cell, or something like that. So there are error rates, but there's built-in things that are few. There are much fewer than happens in computers. And when they happen, there's also things that it may not mean anything. When we get later on, you'll find out when they read codons. When you read codons, that ends up coding for, so you got 3 base pairs coming together. When you read the codons, and this 3 base sequence codes for an amino acid. What they know is that oftentimes if you mess up the 3rd letter in a codon, it doesn't matter, because it still codes for the same amino acid. There's 64 possible combinations to code for the same 26 amino acids. So there's lots of little fail-safes that are in there. The bigger problem like with this is if the spindle fiber breaks. It doesn't happen a lot, but it can happen. Here's the other good news too. Sometimes mutations are so bad, the cell just dies. It's just gone. So it doesn't persist or manifest in some big lingering problem as well. Okay?
Teachers, please excuse interruption. At this time, the HOSA students that are leaving on the field trip need to be released to report to the front of the gym. Once again, the HOSA students going on the field trip need to be released to meet in front of the gym at this time. Thank you.
Okay, next phase, anaphase. So we still don't have the nuclear membrane right here. And what I'll do is, I'm going to try and show these X. They're going to get pulled away. So watch, double chromosomes, when they get pulled, they're going to get pulled like through a fluid space. So they're going to pull like this, and they're going to bend back the other way. And when they finally pull all the way up, they'll relax a little bit. So I'm going to bend them and make it look like they're bent out this way. Sorry. No, I'm doing the other one. You still have the centrioles, and you still have the spindle fibers attaching and pulling. And there's a little bit of a line that starts to form, like a crease almost, that starts to form during anaphase. Here's the last one too. I want you guys are done. So let's just write this. Spindle fibers. Pull apart. Sister. Chromatids. Each side has identical set. So for like humans at this point, in one cell, you have 46 plus 46, which are your 23 pair and 23 pair. When you're done, put your pencil down. Thank you. There's a decent activity that I do with this. It's with cutout things. Don't have time. We've done a lot of other lab stuff. I'm not going to do it this semester, but it's you cutting out chromosomes. There's 6 pair, so it means you have a total of 12 chromosomes, and we use your whole desk on there. And you cut them out, you fold them up, you go through replication, and you do the whole step-by-step and stuff, and we go, what happens during prophase? All right, set everything up in prophase. All right, what happens in metaphase? Set it all up in metaphase, you do all this stuff, and you fold them over, and they look like one chromosome, and then they're basically folded out like this, and they look like double chromosomes. You got all these cool things in there. But at one moment, you can stop, and you go, look, you're taking up your whole desk, and I have a little bit of chalk that goes in there. You're taking up your whole desk for these 6 pairs of chromosomes, right? At this point, when you get to, especially when you get to anaphase, you have 96 chromosomes in a little tiny thing that is a fraction of the size of that grain of salt. When you guys did the one activity, so you had to estimate how big it was compared to grain of salt. So you have a lot of stuff going on there. The other thing that's happening here too is, I know we're saying metaphase, anaphase, we're going to telophase and all that stuff. It's fluid. It's not like it goes prophase, metaphase. You know what I mean? It's a smooth motion, so it just moves and moves and moves and moves. Today you're going to be frustrated because I'm going to give you some pictures to look at, and you'll go, that looks kind of metaphasey, but it also looks kind of prophasey. And you're like, they have late prophase, early prophase. They have late metaphase, early metaphase, or they have metaphase. You know, you're kind of like, this is the characteristic thing in here. And so I'm going to ask you, make a call, what do you think? Some of you get to and you're like, they're all pulled apart, but then it also starts to make this like cleavage furrow thing that you're going to see in telophase. Just know it's very fluid, and you've got to kind of make a call. On the test, I will say, very deliberate. This, make a very deliberate example of each phase. Okay, so you won't have to worry about it too much. Nate.
What are the dots in the middle?
It's starting to we're going to make 2 cells, right? So it's starting to crease out a little bit. Just crease out. Does that make sense? You'll see when we make the next cell. We got telophase, next one. So now what's going to happen, and this is the easiest one to tell. Do like a double bubble thing. That dot was from where that crease was forming, Nate. This is one of those things when you hear it. Yesterday, of course, I got the giggle. Hey. And it's fine, it's fine. I probably would have done it too. But this is one of those things where you go, oh, that's what that word means. When have you ever seen the meat cleaver? Okay, the big, it looks like an axie thing they use in the kitchen. Meat cleaver, to cleave something means to cut it. So when they talk about cleavage, that's where you have a cut or a separation of two. This right here is called a cleavage furrow.
That sounds very improper.
It isn't. It's a cleavage furrow, okay? And that's where we're starting to make that complete separation right now. So then when you see that, you go, hey, I'm in telophase, real easy. But if you're between anaphase and telophase, there's a couple things that you're going to have to look at and make a decision on, okay? So go ahead and, so here, we'll do this. Telophase, nuclear membrane. Begins to appear. So which is good news. I'm going to draw like dots. So nuclear membrane is coming back. Spindle fibers. Begin to relax. So I'm going to put the centrioles on here. And I'll put some spindle fibers, but they're kind of like detaching and going away. When we talked about the microtubules, being able to move the cell around, there are these protein rods that bind up and then disassemble and bind up and disassemble. The spindle fibers are kind of the same thing. We'll put them all together, use them when we need them, break them down. We don't need them anymore. We can use them for something else later on. The other thing that's happening is chromosomes, or chromosomes relax to chromatin. So now we're going to go back to that little squiggly line stuff, okay? Those little like loopy spaghetti stuff. That's going to be in the middle of each one.
What does that say? Chromosomes relax to chromatin?
Yeah. I know we saw it on the video yesterday. I wanted to show you this real quick because I had it on the bell work. We're going? We're going? There we go. I had this image, and I thought it was kind of cool because you see this double helix and you really get an appreciation of how it is. It's all strewn out like this in the cell, but when we get into going into mitosis, you have to condense it. So you wrap around these things called histone balls. They wrap, they triple wrap, they wrap again, and then they're all compacted into your chromosomes, and you end up having that double chromosome because you have both copies. Okay. I'm going to go and flip this sheet over. Are we done with telophase? Okay. That is mitosis. Prophase, metaphase, anaphase, telophase. That's it. Now you have the bookshelves on the end. Interphase, cytokinesis at the end. Cytokinesis is a squirt of a piece of its life. It's almost nothing. Yes, James.
So this whole process is happening like millions of times per second?
Trillions of times per second.
Trillion?
Yeah, trillions. Because 1 to 2, right now, 1 to 2 trillion of your cells are going through mitosis right now. And then, right now, another 1 to 2 trillion. And it's not happening that fast, but it's like they keep changing. As soon as one's done, the next one keeps going, and the next one goes, and the next one goes. But they don't go in order going, I'm 1,643,000,000, it's my turn. They're just all replacing each other, and it's an average thing, okay? So watch. Cytokinesis is the last one. Cyto basically means cell.
So for cytoplasm, plasm just means like...
Cell plasm. Yeah. So anything that's cyto, you'll see it. Oh, if you ever go to, I'm not going to ask anybody to jump out. I've done it, so I don't really care. If you go to GNC and you want the supplements or any of the powders or any other things, you'll see it on. You'll see the words, the buzzwords that are on like aminoblast. Okay, aminoblast. Amino acids are what are used to make what are all proteins. Proteins make tissues. Cytomax. Cytomax is and cyto is like, what do they call these cool words? Cyto just means cell. Cyto. So there's different ones like that. What?
Just the Latin basis.
Yeah. Kinesis means like movement or action. Okay, so this is it, and we're done. We're done with this basically. We're gonna go back to. We end up having two. Watch cytokinesis, cell membrane slash nucleus. Oh no, cytoplasm. Actually. Separate. Wow. Can't even spell that. separate, and then what you end up with is two new, smaller cells. Put my nucleus in there. And then the chromatin is totally relaxed, so you just have like squiggly lines. Okay, you'll see it a little bit today. I'm going to pull this up really quick so you can see the examples that I had on here. If you want to look at this or you want it, it's on Edmodo, so you're more than welcome to look at it, and it has some more details that are on there. I wanted to make sure they had two different varieties so that different learning styles could benefit from either one if they wanted to.
What do you mean cell membrane / cytoplasm are actually separate?
Now, when telophase is kind of creasing out the cleavage furrow, now they're actually split. So basically the moment, watch, when it goes, that's cytokinesis, then it goes interphase.
So the second it pops.
The second it pops, that's considered cytokinesis. And then, yeah, like that. Then it goes immediately back in interphase. Because the whole time that the chromosomes are in mitosis or in duplication, Anthony, we're teaching, get out of here. Love you too. Hang on. Wyland, hold the question for just 1 second. The whole time that we're in double chromosome phase, you can't read the DNA. So whatever the cell was supposed to be doing can't do anything. It's just focused on reproduction. So I've got to get through that, get done, and then relax so the instructions can be read. Okay? Wyland, question?
Is there another step, like when they form four, is that just
It'll go through interphase. And interphase, what happens is that it goes back doing its job, and it will grow and grow and grow, and if it gets too big again, then it splits again, or if it gets too old. So I wanted to show you, I have this, oops.
Still frozen.
Oh, I froze it, that's why. Awesome. Doubling myself up here. Like I said, this is on Edmodo if you want it. This is what they worked on yesterday. This is interphase, right? So there's interphase. Oh, piece of crap. There's interphase, and now how do these guys compare?
They're smaller.
But they were exactly the same.
Yes.
They're the same otherwise. That's your prophase. That's your anaphase. Telophase. And then the one on the bottom?
Cytophase.
No, cytophase.
Oh, wrong one. Yeah.
Promap. What is this one? They're in the middle.
Interphase.
Metaphase. Metaphase. Okay. So that's what we got. Nate, you can go ahead and stop that.